It is my pleasure to be the student coordinator of this program. I wish you all a fruitful and engaging time. Let's begin our session by invoking God's blessings with a silent prayer. Now, I invite Dr. Anu, head of the department, to welcome the gathering and to introduce the resource person.
Thank you, Angel. Thank you. So good afternoon, all. Resource person of the day, Dr. Mohammad Febin Farook, coordinator of the program, Dr. Annie Febi, DBT-STAR coordinator of the department, Dr. Baby Divya, my dear colleagues and dear participants. I'm happy to welcome you all to the webinar on biomedical research, a broad area of science which involves investigation of biological process, understanding disease causality, and developing effective therapeutics. So with great pleasure, I welcome the speaker of the day, Dr. Mohammad Febin Farook. He was working as a researcher at Cleveland Clinic, Cleveland, Ohio, before the pandemic. I'm having an impressive and extensive CV of Dr. Febin before me. I shall present a snippet from it. He completed his master's in marine biology from CUSAT and PhD in biochemistry from the University of Kerala. His doctoral thesis focused on estrogen receptor activation factor, which binds with a non-classical estrogen receptor. At Cleveland Clinic, Ohio, his work was on developing an in vitro model for cortical dysplasia to study the mechanism of epileptogenesis using rat neurons. During his postdoctoral fellowship at the University of Tennessee Health Science Center, he worked on the mouse model of autism and Angelman disorders. As a postdoctoral fellow at VIB KU Leuven, Belgium, he focused on elucidating the molecular mechanism of myoclonus dystonia. And he's a molecular neurobiologist interested in developing a career in academic teaching and research. With over a decade of experience working in labs around the world, Dr. Febin Farook will be enlightening us on various aspects of biomedical research. On behalf of all participants, we welcome you to this online platform. So I also welcome the coordinator of the program, Dr. Annie Febi, Dr. Baby Divya, the DBT-STAR coordinator of the department. Both of them were actually classmates of Dr. Febin during his PG program at CUSAT. I'm happy to welcome all my dear colleagues and participants to this session. So wishing you all a fruitful session. Thank you.
Thank you, Anu ma'am. Now I hand over platform to Dr. Mohammad Febin Farook, sir. So please.
Oh, thank you very much. Thank you very much for inviting me to give this presentation. So thank you so much for giving me this opportunity to give my presentation before you guys. So my name is Febin. As your HOD actually explained to me, I did my PhD in molecular biology, and then my postdoc experience mainly in disease biology. So let's start once again. So I was telling that I'm going to take this presentation out. Formality totally ഒരു informal വേയിലേക്ക് ആക്കാൻ വേണ്ടിയിട്ട് ഞാനൊരു comedy slide ഒക്കെ ഇട്ടായിരുന്നു. But I already told you, but I think that nobody actually saw it. Okay, so this is what is going to happen. Like, we will have a discussion. So what we're going to talk about right now is biomedical research, funding in biomedical research. Funding in the there is a lot of funding in this field. If you can actually see this, the amount, that is $41.7 billion in US. അതിൻ്റെ തന്നെ ഇന്ത്യയിലെ ഭാഗമായി എടുത്തു കഴിഞ്ഞാൽ ഏകദേശം 600 crores ഉണ്ട് the Department of Science and Technology, and then you have the Department of Biotechnology around 3,500 crores, and then you have the CSIR, നമ്മളിപ്പോൾ ആൾക്കാർക്ക് funding തന്നത് around 3,500 crores. So it's a lot of money here. A part of this amount is actually utilized for doing biomedical research. So what we actually do with the biomedical research, that's what I'm actually telling you. So biomedical research basically that is pure research, and then you have applied research, and then you have the clinical research. Or on the net definition that I have given here, then you can actually see what pure research means. Research is to conduct to increase the basic knowledge and understanding of the life process or the process that happens during the disease manifestation. In applied research എന്ന് പറയുമ്പോഴത്തേനും it is mainly done in more with an application point of view in the near future. തൊട്ടടുത്ത കാലത്ത് തന്നെ നമുക്ക് എന്തെങ്കിലും ആപ്ലിക്കേഷൻ ഓറിയന്റഡ് ആയിട്ട് റിസർച്ച് കഴിഞ്ഞാൽ നമ്മൾ കുറച്ചുകൂടെ അപ്ലൈഡ് റിസർച്ച് എന്ന് പറയും. ക്ലിനിക്കൽ റിസർച്ച് എന്ന് പറയുമ്പോഴത്തേനും ഇറ്റ്സ് മെയിൻലി ഫോക്കസ്ഡ് വിത്ത് ഹ്യൂമൻ സബ്ജക്ട്സ് ലൈക്ക് ഡ്രഗ് ട്രയൽസ്, വാക്സിൻ ട്രയൽസ് ആൻഡ് ഓൾ ദീസ് കേസസ്. ഈ കൊറോണ പാൻഡമിക്കിന്റെ കാലത്ത് നമുക്ക് ക്ലിനിക്കൽ റിസർച്ച് എന്ന് പറയുമ്പോൾ വളരെ പെട്ടെന്ന് നമുക്ക് മനസ്സിലാവും. Now, the information എങ്ങനെയാണ് ഈ ഡിഫറെൻറ്റ് branches in biomedical research pass ചെയ്ത് basically always the information എന്ന് ഈ ഡിറക്ഷനിലാണ് പറയാൻ പറ്റില്ല, but still ഇതിൻ്റെയൊക്കെ ഈ സെപ്പറേഷൻ എന്ന് പറയുന്നത് a thin line ആണ് ശരിക്കും. So usually information passes from the basic research to applied field, because and then you actually take this applied research to a clinical research level where we actually test this in the human subjects. Okay, so pure research അവിടെ നിന്ന് നമുക്ക് directly information clinical research ലേക്ക് മാറ്റാൻ പറ്റുന്നതാണ്. So I told you that there is a scientific methodology for doing not only biomedical research, all kinds of research. Our methodology, different steps on these slides. This is what we all learn from during our plus 2 levels. We make observations first, then ask questions about this, and then make a hypothesis, then conduct an experiment to test the hypothesis, and then draw a conclusion. And finally, what we do is we report our results. So, ഇതാണ് ശരിക്കും ഒരു scientific methodology. These are the 4 steps that we undertake when we do our experiment, when we do a scientific methodology. So, humans case ഈ എല്ലാ സ്റ്റെപ്സും നമുക്ക് undertake ചെയ്യാൻ പറ്റും except for conducting an experiment, because it's really very hard to conduct an experiment in a human subject, right? So what do we do? So there comes the role of animal models. അപ്പോൾ അവിടെയാണ് നമുക്ക് ഈ animal modelsന്റെ ഉപയോഗം വരുന്നത്. So that is an important, very important part of biomedical research. So what is an animal model? അതിനെക്കുറിച്ച് നമ്മൾ അടുത്ത സ്ലൈഡിൽ പറയുന്നത്. An animal model എന്ന് പറയുമ്പോഴത്തേനും it's an animal with a disease either the same or like a disease in the human. അതായത് മനുഷ്യരിലുള്ളത് പോലെയോ അതോ അതുമായിട്ട് വളരെയധികം സാമ്യമുള്ള ഒരു അസുഖമുള്ള ഒരു അനിമലിനെയാണ് നമ്മൾ എപ്പോഴും അനിമൽ മോഡൽസ് എന്ന് പറയുന്നത്. അസുഖമുള്ളവരായിരിക്കും, ബിക്കോസ് അത് കുറച്ചുകൂടെ സ്ലൈഡ് അങ്ങോട്ട് പോകുമ്പോൾ നിങ്ങൾക്ക് അത് കുറച്ചുകൂടെ ക്ലിയർ ആയിട്ട് മനസ്സിലാവും. സോ അനിമൽ മോഡൽസിന് ഒരുപാട് ഉപയോഗമുണ്ട്. അതിലൊരു ഉപയോഗമാണ് നമ്മൾ ഡിസീസിൻ്റെ പ്രോഗ്രഷനും ഡെവലപ്മെൻറ്റും അതിൻ്റെ മെക്കാനിസം ഒക്കെ സ്റ്റഡി ചെയ്യാനായിട്ടും പിന്നെ അതിൻ്റെ ട്രീറ്റ്മെൻറ്റിനൊക്കെ നമുക്ക് ഉപയോഗിക്കാൻ പറ്റും. സോ ഇറ്റ് ക്യാൻ ബി യൂസ് ടു സ്റ്റഡി ദ ഡെവലപ്മെൻറ്റ് ആൻഡ് പ്രോഗ്രഷൻ ഓഫ് ഡിസീസ് ആൻഡ് ടു ടെസ്റ്റ് ന്യൂ ട്രീറ്റ്മെൻറ്റ് ബിഫോർ ദേ വർ ഗിവൻ ടു ഹ്യൂമൻ. Then xenografts means like you can actually transplant tumors from humans to these mice models and then try to treat these tumors in those models, animal models. അങ്ങനെയാണ് നമുക്ക് animal models യൂസ് ചെയ്യാം. ഇനിയും ഇതൊന്നും അല്ലാതെ തന്നെ ഈ നോർമൽ ഫിസിയോളജി പഠിക്കാനായിട്ട് നമുക്ക് ഈ animals മോഡൽസിന് യൂസ് ചെയ്യാൻ പറ്റും. Now the animals used in the biological studies, ഏതൊക്കെയാണ് animals നമ്മൾ ഏറ്റവും കൂടുതൽ biological studies, biomedical studies use ചെയ്യുന്നത്? The first candidate obviously is the mouse, because it's the most favorite laboratory animals. Mouse genes are like 80% orthologous to humans. So that means that whatever results we get is can be successfully actually moved from one model system to another. Say for example, clinical trial humans like വളരെ എളുപ്പം കൊണ്ടുവന്നു. Then it has a similar anatomical, cellular or molecular characteristics of the humans. So ഇതിങ്ങനെ പറയുമ്പോഴും നമുക്ക് പലപ്പോഴും തോന്നാം what is the similarity between the human and the mouse, but it's there. You as a biology students you can all understand it, right? Next is the rat. Rat is also a very favorite laboratory animals, and most rat shares human genes with വളരെയധികം സാമ്യമുണ്ട്. And it also have similar anatomical, cellular and molecular characteristics to humans. Next is the fruit fly, the Drosophila. Okay, this is also a very favorite laboratory animals. Mouse and rat name comparing Drosophila has a very small generation period. and it can be easily maintained in the corner of a lab. ഈ പാറ്റിന്റെ മൗസിനെ പോലെ നമുക്ക് വലിയൊരു ഫെസിലിറ്റി ഒന്നും വേണ്ട, ആൻഡ് കെയർ ടേക്കേഴ്സിൻ്റെ ആവശ്യമില്ല, അധികം ഫുഡൊന്നും വേണ്ട, ക്ലീനിങ് സ്റ്റാഫ്സിൻ്റെ ഒന്നും ആവശ്യമില്ല. So the fruit fly is also a very favorite animal model in this biomedical usage. Then comes the zebrafish. Zebrafish is like kind of like a very fancy kind of an animal model. Actually, if you can see, actually, they are used very well in studying developmental diseases, developmental and disease disorders. So one of the major factor that is makes zebrafish very famous in this research field is that they have a very transparent larvae in which you can actually very clearly see reported genes. Reported genes, a protein we'll tag it with some fluorescent protein so that you can actually see where the protein actually is and how it interacts with the other proteins. Here in this case, we can see different colors, right? Like a blue, green, all kinds of colors. That is actually tagged with different proteins so that you can actually see how this protein interact in these larvae in real time. So that is a very, very fancy model. Then you have other animals too, like rabbits for developing antibodies and all these kinds of pregnancy model like that, the body using. And then you have evolutionary ecological study from embryonic development, like xenopus use a lot. And then you have like the llama. This is a very interesting thing that we actually use it to develop nanobodies. Nanobodies are the recent developments in antibody technology. So isn't it becoming an important animal? Llama. Now, so there are other animals also, but this doesn't come like a slideshow. It's all I'm going to put it on because the slideshow is not working here. Sorry about that. Guinea pig on the C. elegans on the gerbils on the chinchillas on the hamsters. So there are a lot of animals that we actually use in biological cells. Okay, so how do we select an animal model for our research? So there are two broad classes for animal models too. One is like an analog, and that means a similar structure, means having a similar functions. Like structure or ball a similar function category. In this case, here is a penguin's fin, penguin's feet, and a fish fin. Then you have the homologous structure, right? It's evolutionary precursors has similar functions. Humans, intellects, humans, hands, cats, whales, and like a different homologous structures. So based on this, we actually select our model, which model to work on. So once we are actually decided to which animal model to use, actually, so how we actually make this animal a model for our disease. Animals because you have to actually induce a disease in these animals, right? So how do we do that? There are different ways actually we can successfully use an animal model. First one is that induce or experimental model. Animals in a model that is to attempt to reproduce conditions found in the original species. Original species means if a human disease and study, we will try to actually make that human disease possible in an animal model. Now, I will tell you how we actually try to do that in the coming slides. Then you have spontaneous or natural models. Spontaneous disease animals it's humans disease. They will be found in the natural population, and we can use that animal for our study also. This is not very commonly used. Then negative or non-interactive models. So these are like a kind of control animals. Control animals, for example, I can give an example like if you model a mouse with certain defective genes, not all pups will have carried that defective gene. So there will be a counter population of wild type that we can use as a normal disease models. When we move to the experimental part, I can explain it in a much better way. ഒരു മൗസിന് ഓഫ്സ്പ്രിങ്സ് ആണ് എപ്പോഴും നമ്മൾ എക്സ്പെരിമെന്റിങ് യൂസ് ചെയ്യുക. അപ്പോൾ അതിനകത്ത് ഡിസീസ് ജീൻസ് ഉള്ള പപ്സും ഉണ്ടാവും, നോർമൽ പപ്സും ഉണ്ടാവും. അപ്പം നോർമൽ പപ്സിനെ നമ്മളൊരു നെഗറ്റീവ് ഓർ നോൺ ഇൻട്രാക്റ്റീവ് മോഡൽസ് ആയിട്ട് എടുക്കുന്നത്. To use it as a control, we call usually as a liternate. That is a perfect control. For example, ഇപ്പോൾ ഒരു മൗസിന് ഒരു 10 കുട്ടികളുണ്ടെങ്കിൽ, പപ്സ് ഉണ്ടെങ്കിൽ അതിൽ 5 will be having disease and the other 5 will be normal. So the perfect control for this experiment will be to use the wild type, that is without any defective gene, as a control, and the other one as a model organisms. Then you have orphan models, like animals with no human or non-counterparties. It's just random animals types of diseases. But most of the biomedical researchers are actually focused on the first category, that is we induce an disease in the normal organism, and then we actually study how the disease is manifested in that animal. Okay. So how do we generate an animal model? നമ്മൾ ഇപ്പോഴും ആ ഒരു ഇതിൽ തന്നെയാണ് നമ്മൾ ഡിഫറെൻറ്റ് മെക്കാനിസംസ് ഉണ്ട്. നമ്മൾ സെലക്ട് ചെയ്ത് കഴിഞ്ഞിട്ട് പിന്നെ ജനറേറ്റ് ചെയ്യാനായിട്ട് നമ്മൾ എന്താ ചെയ്യാൻ പോകുന്നത്? ട്രഡീഷണലി വി യൂസ് ഇമ്മ്യൂണോഡെഫിഷ്യൻ മൗസ്, നേരത്തെ പറഞ്ഞതുമാതിരി നമ്മൾ സിനോഗ്രാഫ്റ്റ് ഒക്കെ ഇംപ്ലാന്റ് ചെയ്യാനായിട്ട് ഇമ്മ്യൂണിറ്റി ഒന്നും അധികം ഇമ്മ്യൂണോഡെഫിഷ്യൻ മൗസ് ആകുമ്പോഴത്തേക്കും ഈ ടിഷ്യൂസ് ഒന്നും റിജക്ട് ചെയ്യില്ല. സോ വി ക്യാൻ ആക്ച്വലി യൂസ് ദിസ് ആസ് എ carrier mother to actually do a breastfeeding. Then you actually insert foreign genes into the animals using transgenic techniques. So that's what we call a transgenic animal model. So transgenic രണ്ട് തരത്തിലും നമുക്ക് animals in a classification. One is the loss of function. That means, as the name itself, our gene function completely gone. The other one is like a gain of function. So overexpression of certain genes. So in one case, there is no expression of genes, so complete loss of protein. The other one is there is an increased expression of certain proteins, and so you will actually have a gain of function. Then the loss of function, there are two types, like constitutive knockout and conditional knockout. And constitutive means it's knocked out from the beginning. So so there is a certain limitations for these kinds of knockout, because if if it is a lethal mutation, then the embryos won't be viable, so you won't get any pups. So to just അതിന് പകരമാണ് നമ്മൾ conditional knockout ആയിട്ട് വരുന്നത്. അതായത് if you give a certain conditions, so certain genes are expressed, so that either it will be a lethal one, or so sorry, when you conditionally knock out, when you actually induce certain promoters, that gene is actually dropped out, so you won't have this expression of that gene from that particular phase of life. Gain of function constitute random insertion models. You simply put a foreign gene inside the embryos, and it just goes somewhere and then just incorporate into the genome and start expressing it. Then in knock-in permissive locus model, rather than the specific region later, we can actually target the genes. And then you have again conditional knock-in models and reported knock-in models. Conditional knock-in models means, as I said earlier, that at a certain phase of life, we can actually give a stimulus so that the transgene starts to express. And then you have the reported knock-in model, just as you've seen in the zebrafish examples, right? You can actually tag the protein of your interest with certain reported genes, so that you can actually see where the protein is localized inside the animal model. Then you can have germline genetically engineered mouse models. So you actually manipulate it into the embryo of certain mouse, so that it is a germline mutation. So usually these mutations are carried along generations. പിന്നെ ഇതല്ലാണ്ട് നോൺ ജെർമലൈൻ ജനറ്റിക്കൽ എഞ്ചിനീയറിംഗ് മോഡൽസ് ഉണ്ട്, പക്ഷെ they are usually chimeric models. So you have like a normal one with a genetically mutated one, and you mix the embryos together to get a chimeric model. So chimeric model പ്രത്യേകത എന്താണെന്ന് വെച്ചാൽ it won't be universally expressed in all the cells. You will have a kind of like a chimeric expression ആയിരിക്കും protein. Then there are alternate techniques that we can actually use to put foreign DNAs inside the embryo. So RNA interference is very commonly used, and the CRISPR-Cas system is nowadays very, very commonly used. When zinc finger nucleoside, etc. You can cut the genome in different regions and introduce new genes, or you can take away genes from them. And then you can actually use some transcriptome-based mechanism to insert DNA into the genome. But these things, but this is not very commonly used, not much commonly used in these cells. So, ഇതുവരെ നമ്മൾ പറഞ്ഞത് ഈ ജനറ്റിക്കലി നമുക്ക് എങ്ങനെ ഒരു അനിമൽ മോഡലിനെ ഉണ്ടാക്കിയെടുക്കാമെന്നുണ്ട്. ഇനി അതല്ലാതെ തന്നെ നമുക്ക് other mechanisms ഉണ്ട് how to make animal model. For example, is a radiation model. The radiation, I will talk about this model more in the coming slides. So, ഇത് എന്ന് പറയുന്നത് ഒരു physical stimulus കൊടുത്തിട്ട് you make a kind of like a damage in the regions wherever you want, and then you validate the model and then you do experiments on this. For example, in this case, actually, you use a sublethal dose of gamma radiation. So what happens is you will get some brain deformalities in the pups, because the pups are usually very susceptible, right? So if a pregnant rat radiation the mother will survive, but the pups will have a severe deformality defects in the brain. Then you can also use chemically induced models of bacteria. For example, you can give chlorinic acid to elicit seizures in rats. It can be used for an epileptic model, and then drugs like pentynyl tetrazole, that is a gamma inhibitors, to actually activate the neurons in the brain. Activate the neurons so they are GABA inhibitors. GABA inhibitors means our brain inhibitory neurons arresting. So what happens is our neurons become very hyperactive, so resulting in seizures. So that's also drug induced or chemical induced models we can also do. Then the traumatic brain injury models. So this is typically what you do is you take a rat or a mouse and hit it with what do you call it? Water guns you put a small nozzle here and then you give some pressure, so the water bullets will brain like it. So this is an example of a traumatic brain injury models that we can actually use. So തലക്കടി കൊണ്ടാൽ എന്ത് സംഭവിക്കുന്നു പഠിക്കാൻ ഏറ്റവും best models again in traumatic brain injury. And then this is a typical example of an ischemic stroke model. You actually do an insertion, arteries and veins you can tie the blood supply to that particular region will be lost in the brain. So you can actually use this as a very good model for stroke studies. So other kind of models other than this genetic injury models models. Now let's look into some of these animal models. Now this is this presentation നമ്മൾ ഈ മോഡൽ വെച്ചിട്ട് എങ്ങനെയാണ് ഒരു എക്സ്പെരിമെന്റ് ഡിസൈൻ ചെയ്യുന്നത്, how we actually look into the results of these models work. So I will give you a couple of examples, and let's see how simple I can be in these slides. So take this scenario for example, like you know a gene that causes a disease. Okay, നമുക്കൊരു ജീൻ അറിയാം. ഇത് sorry. Sorry. നമുക്കൊരു ജീൻ അറിയാം, ഒരു ഡിസീസ് കോസിങ് ജീൻ അറിയാം. In this case, actually, this is a UBE3 expression. So ubiquitin protein ligase എന്നാണ് ഇതിന്റെ ജീനിന്റെ പേര്. This expresses a protein called ubiquitin protein, and its function is actually to tag the protein with ubiquitinase so that they will be directed to degradation. Okay, so even the expression. So usually you have a normal range of expression somewhere in the middle, right? So this is a normal range of expression. You will have different sort of mental disabilities. Okay, so in case of UBE3, if the expression level goes a certain level to the very lowest level, you get a syndrome called Angelman syndrome. That is a complete loss of function of UBE3. So the gene is not at all there in those people. And then if the expression is there is a slight expression of UB3 is there, but it's not up to the normal level. then also you get a syndrome called Angelman syndrome, but it's slightly less severe than the previous one, and we call it as a parental uh, UPD kind of syndrome. Okay, so that's how they actually define this disease. And then if you have an increased expression of UBE3A, there are three conditions that it results. One is the Prader-Willi syndrome. It's a maternal disomy. And then you have an autism, that is interstitial duplication. That is, there is again an increased expression of UBE3A. How does increased expression is formed? Because you have a duplication in that region of chromosome. So UBE3A gene രണ്ട് copy ഉണ്ടാവും, so it will be expressing more. And then you have the last category in which there is a very high increased expression of UBE3A, and that results also in autism. So ഈ ഒരു സ്പെക്ട്രത്തിലേക്ക് വരുന്ന, അതായത് ചില സമയത്ത് നോർമൽ UBE3 എക്സ്പ്രഷൻ ഉള്ള ആൾക്കാർക്ക് ഈ ഓട്ടിസം സ്പെക്ട്രം ഡിസോർഡർ കാണിക്കുന്നുണ്ട്. So these categories is actually comes under the autism spectrum disorder. So you have here a gene whose if its expression is actually disturbed from the normal level, you actually result in certain disease. Okay, so let's see how we actually try to model this disease. So we actually try to use two systems in this. One, we used a bat model, and the other one is kind of like a Drosophila. And this study is based on this a single gene called UB3 that expresses protein P6AP. അതിന്റെ ലെവൽ അനുസരിച്ചിട്ട് എങ്ങനെയാണ് നമുക്ക് ഇതിന്റെ സ്റ്റഡി undertake ചെയ്തിരിക്കുന്നത്. So this looks like a very ഇതിനകത്ത് ഈ സൈറ്റിൽ ടേക്ക് ഹോം മെസ്സേജ് എന്താണെന്ന് വെച്ചാൽ, നമുക്ക് ഈ ഡ്രോസോഫിലക്കകത്ത് you can actually engineer the fly in such a way that you can actually either express a UB3A protein, or you can actually shut down a UB3A protein. So here you have like a loss of function UB3A. Loss of function is there the gene is not there. Then you have a system in which you can actually incorporate a human UB3A protein in that Drosophila, and then fly the UB3A then input together. Then you can actually do a mutant UB3A. ഇതിനകത്ത് ഈ മ്യൂട്ടന്റെ പ്രത്യേകത എന്താണെന്ന് വെച്ചാൽ ഇത് സൈറ്റ് സ്പെസിഫിക് മ്യൂട്ടന്റ് ആണ്. So it will actually have only a specific part of the function is cut off. I'm not going to go very details into that slide. So basically നമ്മൾ എന്താ ഇവിടെ ചെയ്തിരിക്കുന്നത് വെച്ചാൽ, we try to express different UB3A in flies and see what happens. Okay, so just imagine that we have two sets of flies in which one we don't have UB3A and with the other one we have UB3A. So അപ്പോൾ നമുക്ക് അതിനെ ഒന്ന് സ്ക്രീൻ ചെയ്തെടുത്ത് കഴിഞ്ഞാൽ, if you can actually this is a real experiment actually done. So what we will do is I will repeat one more time that we have two different sets of Drosophila. One we have a UB3A gene, and the other one doesn't have a UB3A gene. Okay. So adine nammal protein extract cheyidittu, if you can actually run it in a gel, you can actually separate the individual proteins. You can actually see the differential expression based on UB3A regulated proteins, right? So ഏതൊക്കെ പ്രോട്ടീൻ ആണ് UB3A കൊണ്ട് ഇൻഫ്ലുവൻസ് ആവുന്നത് നമുക്ക് വളരെ എളുപ്പത്തിൽ കണ്ടുപിടിക്കുന്നത്. അതാണ് ഇവിടെ ചെയ്തിരിക്കുന്നത്. So first you actually take the head of the Drosophila. So അതിൻ്റെ വേറൊരു പ്രത്യേകതയാണ് Drosophila. When you work with Drosophila, you get thousands of flies. Okay, so you actually put it in ice. They become very fragile, and when you just sieve it, you get all the head separated out. You can actually use a sieve to just take the head, and then you grind it and get the protein extract. And then you can actually separate it based on the pH. So this experiment is actually a kind of 2D separation. So first you separate it based on the pH, and then you use the fraction to use a normal gel base. So the protein will be separated based on the size. Okay, so pH വെച്ച് നമ്മൾ ഫസ്റ്റ് ലൈൻ സെപ്പറേഷൻസിൽ then again the second separation is done by the gel based on the size. So if you can see here in the B panel, you see there are two samples. In one there are differentially expressed bands. See, differentially expressed bands means that this is either influenced by the expression or the lack of UBT. So each protein identical yearly, you can very easily identify the genes that are UBT regulated or there has an impact on this expression, UBT expression കാരണം ആ ഉണ്ടാകുന്ന ചേഞ്ചസ് നമുക്ക് വളരെ എളുപ്പത്തിൽ മനസ്സിലാക്കാൻ പറ്റും. Okay, so we actually do this same sort of an experiment in a different way. ഇത് തന്നെ നമുക്ക് മൗസിൽ ചെയ്യാം. The baby slip was done in Drosophila. Then you can actually do the same thing in mouse. So here you have 3 mouse models system. Maternal deletion, maternal duplication, and paternal duplication. Maternal deletion means you know, like mouse, actually the genes will be not only in mouse, in all organisms, the genes will be contributed from father and mother, right? So there will be 2 copies of genes in everyone else. So maternal deletion means the genes from the mother side is deleted or it has some kind of difference in it. Maternal duplications means that the gene from the mother side is duplicated. There is 2 copies of the same gene. And the paternal duplication means from the father side that you have 2 copies of the genes. So UBE3 I didn't actually say it previously. This gene is actually imprinted in the father. That means if the father supplies the gene, the defective gene, the offsprings won't have disease because the father's genes are usually the UBE3 gene is imprinted. The paternal copy is imprinted. So that's why we mainly use the maternal copies. So this is an example of how we actually validate the model. So here you have the deletion, and then you have the duplication, and then you have the paternal duplication. So if we come to the slide, this is actually a Western blot that actually visualizes the protein. Okay, so you have a wild type. Wild type means an a normal mouse. So genetically engineered mouse a set of offsprings normal. 50-50 chances for 10 pups you will get 5 normal and 5 will be like this is possible. You always say the normal as a wild. So wild type and the expression of the protein is here. So I don't know whether you can actually see the what do you call it? The cursor moving. Can you see the cursor moving? Can anybody reply?
Yes, Febin.
Okay, so ഇവിടെ നിങ്ങൾക്ക് കാണാൻ പറ്റുന്നത് ഗ്യാപ് DH എന്ന് പറയുന്നത് നമ്മുടെ ഒരു ഹൗസ് കീപ്പിംഗ് ജീനാണ്. അതായത് നമ്മുടെ ഹൗസ് കീപ്പിംഗ് ജീൻസ് അല്ലേ, നമ്മുടെ സെല്ലിനകത്തേക്ക് നമ്മുടെ ദൈനംദിന കാര്യങ്ങൾക്ക് വേണ്ടി എക്സ്പ്രസ് ചെയ്യുന്ന ഒരു പ്രോട്ടീനാണ്. There are different proteins. The gap pH is one among them. Then we have beta tubulin, then all kinds of genes that we can use as a housekeeping gene. So what this shows is, we add the protein equal. We can actually use this to quantify the expression of other genes. Within the base, the quantification. So this one is the gene that we actually try to knock out or increase the expression. So here you can see wild type the expression range. Maternal deletion now, you see it's completely gone in both of these regions. So that means the mouse gene expresses a male. Then you have the maternal duplication one. That means from mother side in one gene, it has a 2 copies. So you can actually see an increased expression of UP3 gene in this protein. Okay. So then the third one is paternal duplication. Paternal duplication increased dose its level is always like nullified because it's usually imprinted. So within the quantification graph, so you can actually see if it's if you take the wild type as 100%, you have a very reduced amount in maternal deletion, and then you have duplications actually is like way too high. So it's more than like it's close to 200%. That is double the amount, and then in paternal duplication level, it's close to like normal or like slightly higher than normal. So this is how you validate a mouse model. This is very important that I will actually show you another slides also in the coming presentation. So this is how you actually validate a model after you develop a model. Okay, so now you have this models, right? So what you're going to do with these models? Okay, this is another validation. This is an immunohistochemistry. You can actually see where these proteins are localized in the brain. This is a section of cerebellum. You can actually see these are mainly Purkinje cells in these brain sections. Cerebellum Purkinje cells are കാണുന്നത്. So they are like they are excitatory neurons that is very prominent in the cerebellum. So you can actually see that wild type. You can see the green dots. They are actually the proteins. So ഈ എക്സ്പെരിമെന്റ് നമ്മൾ എങ്ങനെയാണ് ചെയ്തിരിക്കുന്നത് വെച്ചാൽ I think I'll just briefly explain this. So you have an antibody for E6AP and you put an antibody in this section, and then you put a secondary that actually lights up. In this case, it's a Alexa 488 that gives a green signal. Okay, so the blue color in the it's kind of like a counterstaining that gives stains the neurons. So you can actually see here there is a thin green dots around it. These are the ubiquitin proteins expressed in the Purkinje cells. This is the maternal deletion copy. You don't see any actually. And in the maternal duplication animals, you can actually see a much more brighter green signal. That means that there is an increased expression of the protein. In paternal duplication of it's somewhat close to wild type, or you can say like that. Okay, so now you have a valid model, and then what you will do with this model? One of the experimental studies we actually did is we actually looked for monoamine changes in these brains. So monoamine is typically very example because autism spectrum disorders hyperactivity, anxiety is the effects. So we actually looked into many monoamines like striatum. It just serotonin, dopamine levels, and norepinephrine. I'll just simply go through these slides. Okay, this slides only means that you can actually see an increased amount of serotonin or dopamine. Okay, defective ആയിട്ടുള്ള mouse models ഉണ്ട് എന്നുള്ളതാണ് ഈ റിസൾട്ട് കാണിക്കുന്നത്. So there is not much things to discuss in these slides. Just this is what we actually observe. It's increased monoamines in these mouse models. Okay. So another thing that we actually can do with this animal model is once we have this model, some kind of behavioral studies here we did a very interesting studies and ഒരുപാട് behavioral studies ഉണ്ട്. അതിൽ നമുക്ക് ഈ നമ്മൾ ചെയ്ത ഒരെണ്ണം very important ഒരെണ്ണം എന്താണെന്ന് വെച്ചാൽ you can actually just the home cage licking behavior in the mouse. Okay. So the mouse they have to lick the bottle, right? So this is a highly controlled movement that is controlled by the cerebellum. So if you can see actually the mean lick interval, either ഓരോ തവണയും നമ്മൾ lick ചെയ്യുന്ന interval എന്ന് പറയുന്നത് there is significant differences between these models. You can see this is the wild type, and then you have the deletions models. You can see the mean lick interval is very much increased in these models. So ഇതെന്താ ശരിക്കും കാണിക്കുന്നതെന്ന് വെച്ചാൽ നമുക്ക് ഈ ഇഞ്ചിനിരിക്കുന്ന മൗസ് മോഡൽസിനകത്ത് there are certain defective patterns that is elicited in the brain. അതായത് നമ്മൾ എങ്ങനെയാണോ അത് എന്താണ് how we will understand in the mouse perspective. This is what we do actually. Auto testing. We can actually study the gait, like how the mouse walks and mouse will back, and then we can actually do so many anxiety tests like for a chambers experiments, behavior analysis. And I'm not going to go deep into these things. So you can actually see a difference in the duplication mice also. So this also suggests that when you have a very good mouse models, you can actually work so many things with these models. Okay, so നമ്മൾ എങ്ങനെയാണ് ഡിഫക്റ്റീവ് മൗസ് മോഡൽ വെച്ചിട്ട് ഒരു മീനിങ് ആയിട്ടുള്ള ഇന്റർപ്രിറ്റേഷൻസ് നടത്തും എന്നുള്ളതാണ് ഞാൻ ഈ ഡേറ്റാസ് ഈ സ്ലൈഡ്സ് വെച്ച് പറയാൻ ശ്രമിക്കുന്നത്. അതിൻ്റെ ഇത് മെക്കാനിസവും നമ്മൾ ഹൗ വി ആക്ച്വലി interpret our data. Then you actually can look for other gene expression patterns. Here you can actually see different colors. What this is in this slides, പറയുന്നത് എന്താണെന്ന് വെച്ചാൽ നമ്മുടെ ഇതിലൊരു 7 gene expression patterns കാണിച്ചിട്ടുണ്ട്. So you can see there are the different colors. നമ്മളത് increased ആണോ decreased ആണോ നമുക്ക് എക്സ്പ്രസ് ചെയ്യാൻ പറ്റും. ഈ ജീൻസ് ഒക്കെ ഇതിന്റെ കൂടെ റിലേറ്റ് ചെയ്തിട്ടുള്ള എക്സ്പ്രഷൻ പാറ്റേൺസ് ആയിരിക്കും. ബിക്കോസ് ദിസ് ഈസ് എ മോഡൽ ദാറ്റ് ഹാസ് എൻ ഇൻക്രീസ്ഡ് എക്സ്പ്രഷൻ ആൻഡ് വൺ ഹാസ് എ വെരി ഡിക്രീസ്ഡ് എക്സ്പ്രഷൻ. സോ ഇൻഫ്ലുവൻസ് ചെയ്യുന്ന ജീൻസ് ആയിരിക്കും ഇതിന്റെ കൂടെ നമുക്ക് കിട്ടുക. സോ ഈ മോഡൽ നമുക്ക് ഇവിടെ അവസാനിപ്പിക്കാം. Then we'll actually look into another model actually. ഇത് neural tube defect മോഡലാണ്. This is a dystonia mouse model. This is going to be another disease. I'll make it as simple. ഈ dystonia എന്ന് പറയുമ്പോൾ നമുക്ക് ട്വിച്ചിങ്ങും ഇങ്ങനെ കൈയൊക്കെ കൊണ്ട് പിടിച്ചിരിക്കുന്ന ഒരു അസുഖമാണ്. So the most ഇത് സംഭവത്തിലേ ഉള്ളൂ. ഇത് ഉണ്ടാകുന്നത് ഈ ടോർസിന്റെ ഡെഫിഷ്യൻസി കൊണ്ടുണ്ടാകുന്ന ഒരു അസുഖമാണ്. It is a severely debilitating neuronal disorder. So you just have to think in a way that this disease is caused by the mutation in torsinate. So ഇത് ഞാൻ ഇൻക്ലൂഡ് ചെയ്യാനായിട്ട് ഒരു കാര്യമുണ്ട്. കാരണം നമ്മളിത് ഈ മൗസ് മോഡൽ എങ്ങനെയാണ് സ്റ്റഡി ചെയ്യുന്നത് വെച്ചാൽ നമുക്കറിയാത്ത ഈ ടോർസിനെ എവിടെയാണ് ശരിക്കും ലൊക്കലൈസ് ചെയ്യുന്നത്. This is a nuclear envelope protein. Okay? So if you see in the nuclear sections, you can see defects in these regions. അപ്പോൾ നമുക്കൊരു ടോക്സിനെ മോഡൽ കിട്ടിക്കഴിഞ്ഞാൽ ഉണ്ടാക്കി കഴിഞ്ഞാൽ നമ്മൾ ആദ്യം ചെയ്യുന്നത് we have to validate it, right? We have to see if something is really happening there in this environment. So this is a typical example of how we can actually validate the model. So if here you can see the bubble-like structures in the nuclear envelope. So വേറെ ഒരു കാര്യം എന്താ ഇതിനകത്തുണ്ടാവുമെന്ന് വെച്ചാൽ these torsinate losses can cause exencephaly and curly tail. Exencephaly എന്ന് പറഞ്ഞാൽ the brain grows outside the skull. So this it's actually legal. So this is a normal mouse web. You can see the brain is inside the head, but in this case actually the brain goes outside the skull. Okay. And the tail is usually curly. This is also a phenotype that we observe when there is a loss of torsinate gene. Okay. So here slide in our certain times we actually try to rescue this phenotype. Rescue means so if interactive proteins and literature we will actually see what happens when we actually try to manipulate the interaction proteins. So in this case actually this torsinate protein actually interact with another protein called sun protein. These two proteins are actually nuclear membrane proteins. So even the graph torsine wild type exencephaly percentage in the parameter. Then if you take out the torsine completely, the percentage of exencephaly is 39%. Now you will ask why it's not 100%. Okay. So certain genes are not 100% penetrable. അതായത് ഒരു ജീൻ ഡിഫക്ട്സ് ഉണ്ടായത് കൊണ്ട് മാത്രം അയാൾക്ക് അസുഖം വരണമെന്നില്ല. So that's what we call it at the penetrance of the disease. Okay. So if you have this defective gene, still you can be a normal person. So in this case, the toxin deletion causes the exencephaly in 39% of the times. Now, if you actually if you actually delete the one set of sun protein from this, the percentage reduces to 14, and then if you completely knock out the sun protein, the percentage reduces to 0. This is a typical example of how we actually rescue a phenotype by manipulating a non-interactive gene. That's an example of the slide in the result answer. Now, let's go. It's always not a very disease model and you actually look for to try to validate the model. You get a very good results, and then you follow up the experiments with this model. Because it's not always the case. Okay? Sometimes you will actually get many tricky models too. I'll give an example on a mouse model for myoclonal system. So this is more severe than dystonia because dystonia it has it will also have the myoclonus jerk also, and this is actually caused by the mutation of a protein called the sarcoglycan, epsilon sarcoglycan. I'm just going a bit fast because it's already past, right? So ഇതിന്റെയും paternal mode of inheritance ആണ് നേരത്തെ UBT പറഞ്ഞ പോലെ exactly opposite. Okay, so here I just wanted to understand you that this disease, I just want to just think that this is caused by a protein called epsilon sarcoglycan. Okay, so we made a knockout mouse for this epsilon sarcoglycan, and then we do the gene analysis. Gene analysis, we can do RNA sequences and real-time PCR. So this is a small set of genes, but trust me, this goes to nearly 44,000 genes. Just imagine this slides goes all the way to 44,000 genes. Okay, you have the SG sarcoglycan gene here. You can see it is very much reduced. That means completely gone. Even mouse model sarcoglycan gene there is no defect in these animal models. Mouse and the it's perfectly normal. So if you check the rest of the genes, they are all in the normal range. So what does it mean actually? So humans വളരെ ഡിഫക്റ്റീവ് ആയിട്ടുള്ള ഒരു മ്യൂട്ടേഷൻ കാരണം ഉണ്ടാകുന്ന ഡിസീസ് ഉള്ള ഒരു ജീനെ നമ്മൾ മൗസിലേക്ക് നോക്കൗട്ട് ചെയ്തപ്പോഴത്തേനും ഇറ്റ് ഷോസ് എ പെർഫെക്റ്റ് നോർമൽ mouse. So this kind of tricky mouse models are also there. Okay, so if you see it actually here, the same model the Western to see whether the protein is there or not. Okay, so I just want to focus you on this down panel. I don't want to make it much complicated. Here you can actually see the wild type brain and the knockout brain and the wild type cerebellum and the knockout cerebellum. With band that means the protein is there. Either the protein is there or the antibody is not detecting the correct protein. But in this case, actually, see, in the sciatic nerve, the wild type and the band knockout band. In case of lungs also, you can see the wild type and the band lungs and knockout. So that means the protein is detecting something that is true actually for epsilon sarcoglycan. But for some reason in the brain, it shows a signal in the same our size which non-specific signal correct size epsilon sarcoglycan range. So what we actually can do in these situations like this, we will try to do a different antibody, then maybe we can actually try to develop our own tools. Here in this case, we actually developed some nanobodies. Nanobodies sadhana antibody conventional antibodies size. So this can actually go into regions of the epitopes where the normal proteins don't recognize. So the option epitope antibody protein recognize certain epitopes bind with time. Antibody if it's small nanobodies, it can actually go into different groups and something, and they can actually bind to different regions. We try to develop nanobodies. You can see actually here. So once the nanobody is developed, so then we actually successfully found, we are lucky enough to find one nanobody that actually detect the protein in the wild type, but not in the knockout. So this one is kind of like you can actually see the total protein means this is total protein from the sample, and then you can actually use a conventional antibody. The wild type brain knockout by signal antibody. Then you actually go to this part where you use a nanobody so that it actually gives a blank region. So further experimentally, nanobody we can actually work on these animals, but we have to see what really happens in these mouse models, because the results are not very much satisfactory. Model validate results in the it's highly tricky. This is an example of the like it's always not a smooth path ahead. So now this is something that I wanted to discuss you. ഇതുവരെ നമ്മൾ animal model ഒക്കെയാണ് പറഞ്ഞത്. ഇതും ഒരു animal model തന്നെയാണ്, but this is kind of like from a model you take up the neurons and then do something that is can be easily done. For a rattle mouse like experiment ചെയ്യുകയാണെങ്കിൽ it takes some time, right? So and also the duplication purpose നമ്മൾ ചെയ്യുമ്പോഴത്തേനും the rat mouse costs a lot of money because you know 6 animals we need 6 lakhs. So here let's see how we actually try to do it in a much cheaper way, effectively. So this is an induced model example. So how we induce a certain disease in rat models. So this is a rat model for epilepsy. Let's see how we did it. So as I said earlier, so ഇതിനകത്ത് നമ്മൾ cortical malformation disease model ആണ് develop ചെയ്യാൻ നോക്കിയത്. So the disease is called cortical dysplasia. So how we try to model the animal is like you irradiate the rat, the mother, with sublethal dose of gamma irradiation, and then you wait for the pups to be born. അപ്പോൾ ഞാൻ നേരത്തെ പറഞ്ഞതുപോലെ ഈ റേഡിയേഷനകത്ത് ഏറ്റവും കൂടുതൽ എഫക്ട് ചെയ്യുന്നത് പ്രഗ്നൻ്റ് ആയ മൗസായിട്ട്, കാരണം അതിൻ്റെ എംബ്രിയോസിനായിരിക്കും. So you validate the embryos for what really happened. So here is it's a typical validation slides. So you can see controlled and irradiated brain sections. Visibly കണ്ട് നമുക്ക് ഒരുപാട് ഡിഫറൻസ് ഇതിനകത്ത് കാണാൻ പറ്റും. സ്മോളർ ബ്രെയിൻ ആണ്, cortical structures ആണ്. And if you look deeply, you can see neuronal clusters and thin cerebellar cortex and the hippocampal crest. These are the typical features of cortical dysplasia in humans, right? So we are trying to model a cell culture system for cortical dysplasia. ഒരു ഹ്യൂമൻ ഡിസീസ് നമ്മൾ മോഡൽ ചെയ്യുമ്പോഴത്തേനും നമ്മൾ ഇത് ചെയ്തിട്ട് നമ്മളത് വാലിഡേറ്റ് ചെയ്യുമ്പോഴത്തേനും we get somewhat a very similar features. So they also have low threshold for PTC generation. That means e mouse model stereo dose GABA inhibitors the rat starts to seizure. So they also have spontaneous seizures. Normal drugs they will have spontaneous seizures. So this is the slide that shows the histopathology in humans. Actually, you can see the control animals. The typical pattern in the cortex layers, brain, there are different layers, 6 layers in the human's mouse. All we have 6 layers, right? So in the focal cortical dysplasia sections, these are a pattern come to hold. Also, you will find so many dysmorphic neurons, bubble-shaped neurons. Our disease in the typical feature. You see the same thing in the irradiated models of rat too. So, ഇങ്ങനെ ഒരു റാറ്റ് ഉണ്ടാക്കി കഴിഞ്ഞാൽ നമുക്ക് നമ്മുടെ എക്സ്പെരിമെന്റിനെ എങ്ങനെ എക്സ്പെഡൈറ്റ് ചെയ്യാമെന്നുള്ളതാണ്. സോ can we do it in an in vitro model? In vitro model means നമുക്ക് ഈ റാറ്റിനെ തന്നെ യൂസ് ചെയ്യാണ്ട് കുറച്ചും കൂടെ ഈസിലി ആക്സസിബിൾ ആയിട്ട് കുറച്ചും കൂടെ മാനിപ്പുലേറ്റ് ചെയ്യാൻ പറ്റുന്ന രീതിക്ക് നമുക്ക് ഈ സെൽസിനെ ഐസൊലേറ്റ് ചെയ്ത് ചെയ്യാമെന്ന് we tried this. Okay. So what we did is, as I said, we give the mother the irradiation with sublethal dose. In E17 on the day, because one important factor is the experiment that we have to have a repeat means protocol. If you do an experiment at a certain phase of time, you have to keep on doing it at the same time, right? So here we actually irradiated on day 17. And then on day 20, we we started isolating the neurons, and we grow the neurons for 15 days in the plate. So this is how a neuron looks like. I think I have a slide. So this is the progression of growth of these neurons. You can see from D5 to all the way to D18. But in neurons and cultures in the when they become day 15, they are completely matured in the sense that with matured neurons and the LR property, it will start showing on day 15. So we can actually do any sort of experiments on day 15. So now we have the animal model and we validated the model. Now we actually isolated the neuron from these models. ഇനി നമുക്ക് അതിനകത്തേക്ക് എക്സ്പെരിമെന്റ് ചെയ്യാൻ പറ്റും. So we first looked into this. This looks like a very complicated, but I'll just simplify it. ഈ പാനൽ മാത്രം നോക്കിയാൽ മതി. You have the normal rat and normal neurons, and then you have the exciting neurons. ഇതിനകത്ത് കാണുന്ന ഓരോ dark red spot, it is a synaptic connection that is formed from other neurons. ഇപ്പോൾ കൾച്ചറിനകത്ത് നമ്മൾ സിംഗിൾ ന്യൂറോൺസ് അല്ലല്ലോ ഉണ്ടാവും, എല്ലാ ന്യൂറോൺസും ഉണ്ടാവും. So in this case, actually, you can see in the excited panel, in the last slide, day 15, you see a lot of dark spots, right? These are all neurons. These are all regions where this neuron communicate to other neurons. So you know what an epilepsy is, right? Epilepsy is kind of like you have a heightened reaction in the brain. So ഒരു data sheet ഇങ്ങനെ കാണിക്കുന്നതിനേക്കാളും നല്ലത് എനിക്ക് തോന്നുന്നു if you actually see what happens in an epileptic brain. So this is actually an EEG of an epileptic brain. Okay? This is a normal brain. It's a normal brain. This is how the wave goes in a normal brain. So this is what it happens when we start to seizure അടിക്കുമ്പോൾ നമ്മുടെ ബ്രെയിൻ പാറ്റേൺസ് എങ്ങനെയാണ് മാറുന്നത് എന്നുള്ളതാണ് ഈ EEG കാണിക്കുന്നത്. This is just the start, and it finally goes to something like this. It's totally crazy. Okay. So അതിൻ്റെ ഒരു ടിപ്പിക്കൽ എക്സാമ്പിൾ ആണ് നമ്മളിവിടെ കാണുന്നത്. അതായത് ഈ ന്യൂറോൺസിനകത്തുള്ള ഓരോ സ്പോട്ട്സും ഇറ്റ് ഈസ് ആക്ച്വലി കമ്മ്യൂണിക്കേറ്റിംഗ് വിത്ത് അതർ ന്യൂറോൺസ്. ഇഫ് ദിസ് വൺ ഈസ് നോർമൽ, ദിസ് ഈസ് ഹൈലി അബ്നോർമൽ. സോ ദിസ് ഈസ് വാട്ട് ഇതിൻ്റെ ക്വാണ്ടിഫിക്കേഷൻ ആണ് ഈ കാണിച്ചിരിക്കുന്നത്. You can see a very increased amount of neuronal connections in these neurons. So, okay. So, and also we can actually see these formations. This is a very young neuron, like it's day 2 neurons. So you can actually see this heightened region. These are all regions of pilopodal growth. Pilopodal growth എന്ന് പറയുമ്പോഴത്തേന് it is actually actively seeking other neurons to communicate. You can see the difference between the normal and the excited means നമ്മുടെ disease models neurons. So this is how the neurons actually grow in these excited neurons, in these excited axons. Usually looks like a spine. നമ്മുടെ ന്യൂറോണൽ കൾച്ചറിനകത്ത് ഒരിക്കലും ആക്സോണിൽ സ്പൈൻസ് കാണില്ല. എപ്പോഴും the spines will be always in the dendrites, right? Dendrites are here, but these are axonal spines. These are not spines actually. These are all pilopodias in which the neurons are actively looking for others to communicate. So there is very much defective in these neurons and not the കാണിക്കില്ല. Okay, so yeah, I think this is going to be. Oh no, I have one more slide. I think I'm missing that slide. Okay, sorry. Let me check. ഒരു slide കൂടെ എനിക്ക് കാണിക്കണം ഇതിനകത്ത്. I somehow missed that slide. I'm taking a lot of time. Okay, so this one I wanted to show you that this is the slides I missed in the previous one. So ഇതിനകത്ത് നമ്മൾ നോക്കുകയാണെങ്കിൽ this is a typical example of the rescue of that property. നമ്മൾ നേരത്തെ പറഞ്ഞ neuronal property നമ്മൾ എങ്ങനെ rescue ചെയ്യാം by adding certain drugs. This is the same results. Here you can see baseline എന്ന് പറയുമ്പോൾ നമ്മൾ normal expression ആണ്. അതിനകത്തേക്ക് irradiatedനകത്ത് increased synapses formation. Synapses formation means the active regions where the neurons communicate. And then you have when you give a certain drug, it increases stimulation. Then you give certain drugs, the synaptic pigments comes to the normal region. Certain drugs. In this case, NMDA receptor analogs it comes to the normal region. When you block the NMDA receptor, it comes to the normal regions. Okay. So this is a typical example of how we do the rescue in these types of studies. Yeah, I think probably that will be all I have to present today. And this is the group that I work with. Last thing I know, but this is the lab I worked with actually. And thank you for listening. I think this is going to be my thank you slide.
So with that, we have come to the end of our webinar. I once again thank our resource person, Dr. Mohamed Febin Farook sir, faculty coordinator, Dr. Annie Febin ma'am, and all others who made this event a great success. Thank you all.